This video is about enzymes. Enzymes are catalysts, which we've talked about in year 12 when we did rates of reaction. Catalysts speed up the rate of a reaction by reducing the activation energy. Enzymes are amazing catalysts. They can speed up reactions by as much as 10 to the 10-fold. They consist of one or more polypeptide chains and may also include a cofactor, which is usually a metal ion or a small organic molecule. One characteristic of enzymes is that they are extremely specific. That means that they only act upon certain substrates, and a substrate is a compound that the enzyme acts upon. Part of the enzyme that's responsible for its catalytic activity is called the active site. The active site may only be a small part of the overall molecule. It's usually a cleft or crevice on the enzyme's surface, and it contains groups from amino acid residues that interact with the substrate, binding it in place, and which catalyse the reaction. The way that the enzyme works depends on its precise three-dimensional shape. Only substrates with the correct or complementary shape fit into the active site and bind to the enzyme. Enzymes are so specific that they can even tell the difference between different stereoisomers, so that only one enantiomer of an optically active compound will bind to the enzyme and the other enantiomer won't. This is known as stereospecificity, and it's extremely useful to chemists trying to synthesize chiral compounds. Stereospecificity occurs when only one enantiomer of a substrate has atoms in the correct position to interact with groups on the enzyme's active site. Substrates bind to the active site in a number of different ways. They involve intermolecular forces and ionic bonds. The diagram shows the lock and key hypothesis, where the substrate and enzyme have complementary shapes and they fit together perfectly. A shortcoming of the lock and key hypothesis is it doesn't explain how the enzyme catalyses the reaction. So the model was extended by the induced fit model. In this model, the substrate binds to the active site, inducing a change in the shape of the active site to allow the substrate to fit perfectly. Bonds are made between the enzyme and the substrate, which promote the movement of electrons within the substrate, making it easier to break and form bonds. This lowers the activation energy for the enzyme catalyzed reaction, and the model is shown in the diagram. So the hexagon binds into the enzyme active site, induces a change of conformation. In the transition state, the bond between the hexagon and the pentagon is strained, and this makes the activation energy for breaking the two parts of the substrate apart lower, and once the reaction has occurred, the products diffuse away from the surface of the enzyme, which can then catalyse the reaction of another substrate molecule. Lactate dehydrogenase is an enzyme. It catalyses the oxidation of lactic acid or 2-hydroxypropanoic acid. This is formed in your muscles when you exercise anaerobically, like if you complete a 100-metre sprint. It's what makes your muscles ache. The lactic acid has to be converted into another chemical before it can be processed by the body. Lactic acid has two enantiomers because it contains a chiral carbon, one that is attached to 4 different atoms or groups. Draw the two enantiomers of lactic acid. Look at the two enantiomers and check that they are mirror images of one another. It's best to add the groups attached to the chiral carbon in pairs. So you add the 2 methyl groups at the top first, and then the 2 hydrogens nearest the mirror plane, then the 2 OH groups, and finally the 2 COOH groups. Remember to check that you have the connectivity of the atoms correct, so that the carbon is joined to the oxygen of the OH group. Lactate dehydrogenase is stereospecific, and the active site can only bind one of the 2 enantiomers. The diagram shows the side chains of the amino acids that interact with the substrate, and you can see that the substrate is held in place by hydrogen bonds from 3 different amino acids. The other enantiomer of the substrate would not have the OH and Cu groups in the correct position to form hydrogen bonds with the amino acid side chains of the protein. Enzyme inhibitors are molecules that stop enzyme catalyzed reactions taking place. Inhibitors have similar shapes to the substrate. They're important molecules and are often used as drugs to treat disease, where they interact with enzymes that are involved in the disease process. The inhibitor makes similar bonds and interactions with the enzyme as the substrate, but it can't react. This type of inhibition is called competitive inhibition, with the inhibitor and the substrate competing for the active site. The amount of inhibition depends on the relative concentrations of the inhibitor and the substrate. About 500 different targets have been identified for drugs to treat disease in the human body. Many of these drugs target enzymes. They act by inhibiting the activity of an enzyme that catalyzes a harmful reaction. Examples of enzyme inhibitors include ones that stop blood clotting, the protease inhibitor that stops the HIV virus from replicating, and inhibitors to stop enzymes releasing too much acid in the stomach. Historically, scientists discovered new compounds, often from natural sources such as plants, and then used experiments to work out how they could use the newly discovered compound. Nowadays, though, a more technical approach is adopted. The structure of enzymes can be determined using techniques like X-ray crystallography and nuclear magnetic resonance spectroscopy. It's then possible to design drugs that will fit into the enzyme's active sites using computer modelling. This is known as structure-directed drug design. Chemists use computer modelling to design molecules that will inhibit enzymes known to be involved in diseases. One of the first designed drugs was Relenza, which prevents influenza infection. Scientists determined the structure of the neuraminidase enzyme, which is involved in the infection of the flu virus, and then designed Relenza to stop this enzyme working. The molecule is shown in the diagram. Stop the video and work out how many chiral carbons there are in the Relenza molecule. Also identify which functional groups are in the molecule.